for what it is. It's a troponin. The second one tells you its function. Because this TC one right here has a calcium binding site. Does that make sense? Yes.
That last T mix.
So,
Topamine?
topomyosin, interaction, calcium.
And the first T stands for what?
Troponin, because this is troponin. So troponin T, troponin I, troponin C. But what does the C, I, and the T stands for? Tropomyosin, interaction, and calcium. Because that part, the last part tells you its function. Now in here, the structure, cut it in half. So this part is only going to show the TTC. This is going to show you the thin filament collectively. The complete thin filament, and this is just going to show you the TTC by itself. Does that make sense? So we have the tropomyosin. So it's harder to draw, but the TC part, the TI part, and the TT part. Does that make sense? Or does that look confusing? Let me draw it. I'm going to draw the same thing, but because I can't, I'm going to draw it up here because it's very hard to align stuff over here. So I'm going to draw the same thing over here. Okay? So you have your tropomyosin. Attached to it, you have troponin. So this is the TC, TI, TT. Does that make sense? And this is the calcium binding site. Does that make sense? Yes.
Are they always in that order?
Yes.
Okay.
So TC is always on the outside.
Yes. Because this is why it's TT, because troponin is bound to tropomyosin. Okay? TI is the interaction between the TT and the TC. And the TC is always facing the exterior because that's where calcium will come and bind. So it cannot be opposite.
So it only enters into that little site thing once from the top, only not in between, like the TC, then the rod, the calcium binding site, and then.
No. So calcium binding, it's basically, this is my, let's say I have 3 chairs, okay, stacked upon each other, okay? The one that is touching the floor would be like the TT, okay? The one in the middle that's touching the boat would be the TI. And would you be sitting on TI or TT or TC? TC. Does that make sense? Okay? So I could only sit, and I have 3 chairs stacked up, but I could only sit in the topmost one, which I would be calcium in that. Okay? Okay? You seem confused.
Well, I was good until the calcium's touching the TT.
So the T, not TT, TC.
Oh, okay.
I just said, would you be able to sit on a TT?
Oh, okay.
If I have 3 chairs stacked, would you be able to sit on the lowest one? No. Does that make sense? Okay?
And, sorry, and
It's okay.
just to clarify, and the line that the TT is touching, that is the myosin.
Yes. Okay? So you have tropomyosin, you have troponin has 3 segments to it, okay? But this entire thing is one troponin molecule. It has 3 different segments. It has one segment that is directly touching the tropomyosin. This is why it's called the TT. It has one segment that actually binds calcium. That's why it's called TC. And then the middle segment, which connects the 2 other segments together, and that's why it's called TI, because it allows interaction between the 2 other segments. Make sense? Okay. So this together is the TTC complex. Troponin-tropomyosin complex. Okay? Now, what did I say thin filaments were? Actin chains plus TTC. So both of that is put together to make thin filaments. Actin chains plus TTC. So that's what, I'm going to erase this, and I'm going to draw it again here, but you guys should draw it in this space. Does that make sense? I think I might have a different slide. Let me check. Okay, I do have a different slide for you guys. No. I have a different slide where it shows it with thin. So the heading should be thin TTC or thin filaments. So it's thin filaments plus TTC. So the heading of that slide. So where do you find thin filaments? So the heading should read thin filaments. I used to explain it a little bit differently, and I found this way is much better, but I just haven't gotten around to changing my notes. Okay. So, where do you find thin filaments? Talk to me. Talk to me. Underneath the I band and the A band, right? Because A band is the interaction between the two. Now, structure. We're going to draw the structure of complete thin filaments with the actin chain as well as the TTC. So let me draw one thing at a time. Don't draw and use different colors. I'm going to try to use as many different colors as I can to color coordinate stuff. Go ahead and draw this. Now, on the side, do not label it on here. That's why color coordination, so write it somewhere. We're going to write something else here, but for me, I'm writing it right here because I can erase it. Okay? This is, the red is what? What is the red? Nope.
Tropomyosin.
Nope. Actin chain. Actin chain. What is the green? Myosin. Binding site on actin. Myosin binding sites on actin, right? well, pretend this was a highlighter. What am I talking? It's supposed to be thick, like a highlighter would cover the entire thing. You need to use those colors, just something different.
I just said.
Okay. Okay. So the blue is what? Huh? Tropomyosin, good. Tropomyosin. Blue is tropomyosin. What is this? Troponin with its 3 segments. 1, 2, 3. Okay? Troponin with 3 segments. I can't seem to align this. And what is this red dot on this last one? Calcium binding site. Calcium binding site. Does that make sense? So, this collectively is a TTC, correct? So, doesn't thin filaments have TTC plus the actin chains? So here is the tropomyosin. Here is the troponin with the calcium binding site. Make sense? Now, what do you think the function of TTC is? Of TTC. I'm going to talk about the function of TTC. No? What do you think the function of TTC is? Look at how it's oriented on the actin chains. What is so significant? It's particularly oriented in a specific way. What about the How is the TTC oriented on actin chains? What do you see? Hmm? Okay. Now this is going to be a demonstration which you guys don't start laughing. I need a guy and a girl to come up. Come on, volunteers. One guy, one girl. I'm going to start picking you guys, so hurry up. Come on. Yes, you are. Just be careful with this. Okay. Sorry you guys for coming. Okay. So let's say they're at a dance. These are two partners, right? They're going to dance, right? Now let's say I'm another girl So I kind of push her aside and I dance with him. What am I doing?
Interfering.
Who am I? Am I interfering? So what am I inhibiting?
Their interaction.
Blocking, I'm interacting, right? I'm blocking what?
Binding side.
Exactly, what binding side? Because if I'm here, can she dance with her partner? No. So who am I? Yes, according to this photo, whatever. Does that make sense? So let's call her actin. Let's call him myosin. Okay, so actin and then myosin. When the thick and thin filaments interact, they're contraction. So when they interact, there's dance, right? Now I come in and I block it. Can they dance?
No.
No. So who am I?
Tropomyosin.
TTC. What part of TTC? The topomyosin. Because do you see, you guys can take a seat. Do you guys understand the example?
Yeah.
Okay. So TTC, do you see the topomyosin part is actually closing up the myosin binding sites on actin? There, if this is my myosin binding sites, and if I close it up, can myosin binding sites bind like that right now? No, because the topomyosin is closing it up. Make sense? Yes? So what do you think the function of TTC is? What is the consequence of that? Why? What is the function? Huh? Inhibit or prevent muscle contraction. So what is the function of TTC is to prevent muscle contraction. Function of TTC is to prevent muscle contraction.
Who's the guy in this there?
He was the myosin, and she was the actin chains, and I was the TTC. To be more apparent, the tropomyosin of the TTC, not tropomyosin. Does that make sense? Yes? I mean, I block the thick and thin filaments from interacting. Well, I mean, it covers up, because this is where myosin would bind, right? So it covers up the myosin binding sites. Okay? Well, maybe I should have made her myosin, I should have made him actin, but anyway. But the point is the TTC are overlapping the myosin. They're closing up the myosin binding sites. If the myosin binding sites are closed, myosin cannot come and bind them. If thick and thin filaments don't interact, then there's no muscle contraction. Make sense? Yes? So the function is to prevent muscle contraction from occurring. How? How does it prevent muscle contraction? Well, it closes up the myosin binding site, so myosin cannot bind with actin. If myosin and actin don't bind, they don't come together, there's no muscle contraction. Does that make sense? Yes? Yes.
So, for it to attach the binding site, troponin and tropomyosin, they have to break. For it
Okay,
to-
say that again.
So for that,
For tropomyosin to cover up myosin binding sites?
yeah, something to attach the binding site, so they have to break the
The-
tropomyosin and the troponin.
No, no.
Because together
They form the TTC. So TTC covers up the myosin binding site. What part of TTC actually closes it up is the tropomyosin. I'll tell you what the function of tropon is in just a second. Exactly, not allowing actually actin and myosin to interact. Myosin is thick filament, thin filaments are actin and TTC. Yes. Well, the red and the green is, there's no myosin drawn here. No. Binding site on it. I have not drawn thick filament. There's no myosin. So quiet. Remember I said thick and thin filaments have to interact with one another to cause muscle contraction? Thick filament is myosin molecules. What part of actually thin filaments? Actin. Because thin filaments is actin and TTC, right? So when myosin and actin bind together, there's contraction. What is the function of TTC? Is to close off myosin binding sites on actin, therefore actin and myosin cannot come together. Does that make sense? But you always say, because actin is still thin filaments, right? It's a part of thin filaments, but it's still considered thin filaments. Does that make sense?
Is there anything that within the thick filament that's spontaneous?
Yes, you had a question?
Does the troponin have a specific function?
Yes, which we'll get to.
Okay.
You guys understand this? Yes.
So it's not actual myosin, it's the binding sites
What?
that it interferes with, like it blocks it.
Yeah, it doesn't block
Actual
myosin.
myosin, it's just the
It
binding
blocks a
site.
place where myosin will come and
Oh, okay.
attach.
Okay.
Okay? It's like an example. I was not blocking her. I was blocking her position on him.
Okay.
Okay? Make sense? Yes.
So, thick and thin filaments cause contraction.
But they interact with each other. Yes.
When they interact. And then, so with the TTC, cause the release?
TTC just prevents the contraction from occurring. Can you say that it actually causes relaxation?
Yeah.
In indirect way. Because something else actually causes relaxation. It just prevents muscle contraction from occurring.
But how could the relaxation is not contracting, so there's
See that?
you said it's a form of relaxation. So how can you relax them when they're contracting? They don't bind, they don't contract.
But at one point they were binding.
Okay.
Okay, there were one point they were binding, which we have to get into detail to actually show you what's going on. You'll understand once the entire story is told. Yeah. Thin filaments is made up of actin chains plus the TTC. Now, so in this photo again, A was actin chains, C was myosin binding sites on actin. Now, what is B? B is tropomyosin. D is troponin. Do you see that how the rope is on the site of this myosin binding site, so the myosin binding site cannot come and bind them? Do you guys see that? Okay. Now in this image, what is E? These spaces right here are calcium binding sites. So these sites right here on troponin are the calcium binding sites. I mean, it has more, but you don't need to know how many. Okay? Does that make sense? So as you can see, when the TTC is moved over, you expose the myosin binding sites. No, this is the TTC, this is troponin, the green would be the calcium. Okay. So what allows myosin binding sites to be exposed? So basically, think of it this way. This chair is actin, one of the actin molecules. You have multiple chairs. Okay? Right here is the molecule, and this area is where myosin come and bind. Okay?